What I'm going to be talking to you about is regenerative patch technologies and our development of a composite Rpe cell ultra-thin Perylene implant that is used for the treatment of geographic atrophy. And I'm Jane Lebkowski. I'm the president of R&D of Regenerative Patch. So just a little bit of background on regenerative patch. We call it RPT. RPT was formed actually in very late 2011, and with founders who are the key developers of the technology, Mark Humayun, Dennis Clegg, and David Hinton from USC, and also from Caltech and from University of California, Santa Barbara. And they are the founding technologies that have been gone into developing the product that I'm going to be talking to you about. And RPT has an exclusive license for this combined technology from the three universities to be used for the development of our implant for the treatment of geographic atrophy. So there's a whole host of people now who are involved with RPT, including the founders, myself, and several people also from the cell and gene therapy world who have been involved in development of other types of therapies. So the program really is, our first product is really targeting geographic atrophy. Geographic atrophy is the later stages of development of a disease called age-related macular degeneration. And what happens in age-related macular degeneration is that in the early stages of AMD, what you have is dysfunction of the retinal pigmented epithelial cells, you have the beginning of degeneration of the Bruch's membrane, and the accumulation of drusen. And this gradually progresses, it almost inevitably progresses to the advanced forms of AMD. One of them is called wet AMD, in which you have the wet or exudative form of AMD. And then there's also geographic atrophy, or the dry atrophic form of AMD. About 80 to 90% of patients who suffer from AMD have the dry form. Wet AMD, there's about 10 to 20%, and there are effective drugs out there right now, Lucentis, Eylea, others that are really targeting to destroy the neovascularization that happens in the choroid in wet AMD. But there's nothing out there that is approved for, the dry form of AMD. There are nothing. The only other possible therapeutics are looking at preventing the progression of AMD, and those are mostly through vitamin supplements. Nothing has proven to be really effective in the treatment of AMD, or even really slowing its loss. So what happens here is that in geographic atrophy, you have a loss of photoreceptors and extreme loss of vision loss. So it's really the technology that came from the three laboratories that I'm talking about, which is to really look at replacing the damaged Rpe cells and the Bruch's membrane, which also is damaged in patients with age-related macular degeneration, and in particularly dry AMD. So this is our product. It's called CPCB RP1. It is a composite Rpe cell Perylene membrane implant, just diagrammed here. And essentially what it has is an ultra-thin Perylene membrane that allows for the diffusion of macromolecules very much across that particular membrane. And it is machined in such a way, it's diagrammed here, and you can actually see it. It's machined with ultra-thin and a support structure of a mesh. And it allows for the diffusion of macromolecules across that particular membrane, very similar to the types of diffusion that is seen in the Bruch's membrane, the native Bruch's membrane itself. Similarly, on top of that Perylene membrane are Rpe cells. In this case, Rpe cells that are derived from human embryonic stem cells. And it was this technology that came from Dennis Clegg's lab. And this is a picture of the implant. Essentially, it's approximately 3.5 by 6.5 millimeters in area. Has a little tab that's designed and a little what we call the nose. And the nose is to provide for directionality and allowing the surgeon who is implanting it to actually orient it in the appropriate way. And the intentions are is to use this implant to interdigitate between the photoreceptors and the choroid to be placed in the subretinal space to essentially replace what is damaged in AMD. So again, I told you that there are two very critical components that go into the manufacture of this particular implant. The first is the Perylene membrane itself. It's manufactured from Perylene, Perylene C, and it is a USP Class 6 biopolymer. So again, has been used in a variety of different implants implants that are used routinely for the last 30 years in biomedical sciences. And also, it can be machined to very, very, very precise thicknesses using MEMS technologies to allow for precise diffusion of macromolecules across that particular membrane. And this is just some data that came out of Caltech to show that the different permeabilities can be used to mimic what that is that of the Brooks membrane. The other critical component is the Rpe cells themselves. They're derived from pluripotent stem cells, specifically human embryonic stem cells. And these are differentiated. They're approximately 99% pure Rpe cells, and they have all the various functions of Rpe cells. They produce pigment. They polarize on a Perylene membrane, and that is one of the important features of why we want to have this Perylene membrane, to allow the Rpe cells to polarize. In many cases, the Brooks membrane is defective in patients with dry AMD. And as a result, even if you seed cells on top of it, they have nothing to anchor themselves on or nothing healthy to anchor themselves on. So in this particular case, we're using the Perylene membrane to allow these Rpe cells to polarize. We know that they form tight junctions, which is a critical feature of the Rpe cells. And importantly, they secrete from their apical and basal surfaces macromolecules that are important. So, for instance, for PEDF, that is critical important on the apical surface to support the survival and function of photoreceptors. And on the basal side, VEGF, so that you can complete and actually look at survival and the health of the vascular system below below the subretinal space. So what you can see here is just a picture of these Rpe cells with the numerous villi on the apical surface that really allow them to integrate appropriately with photoreceptors. The last key step is how do you deliver this particular implant? As I said, it's about 3.5 by 6 millimeters, 6.5 millimeters in area, and it's approximately 6 microns thick. The ultra-thin areas are 0.4 microns thick. And so what we did was design a special tool that can be used to implant this device. In this case, because the Perylene membrane in the implant is pliable, you can fold it up like a taco shell and make it into a smaller cross-sectional area so that you can make a small 1 millimeter retinotomy in which to insert it into the subretinal space. This is just a picture of the tool that was custom designed. And what it does is it has forceps on the end that grab the implant and then protect it while it's being in a shaft, in a cannula, in a shaft. And then that protects the membrane and the implant during the insertion process. So again, it enables very much controlled delivery of the implant. This is just a picture, a video of the implantation procedure. So in this particular case, we're first looking at creating a bleb. There's a vitrectomy that happens, and a bleb is created. In this case, the bleb is created by a hydrodissection, a very careful hydrodissection that occurs to lift the retina and separate it into the subretinal space. Here you can see the tool with its Perylene membrane, the Perylene membrane and the implant inside. You'll start seeing it come out. Here it is, just starting to come out and being implanted into that little retinotomy site that is made. It's being positioned right now in this particular, in the bleb. And you'll see in the next slide, here it is, the implant. And here's the implant here in the subretinal space. So this process takes place in an outpatient procedure. It takes about 2 hours to complete start to finish. So this implant now has been utilized in a phase 1/2A clinical trial. It was an open-label trial in patients with advanced dry age-related macular degeneration with geographic atrophy that involved the central fovea. There were 16 patients who have been entered into the trial. Again, as I showed you, it was delivered using the surgical procedure that we have, that I outlined, and it's one implant. The implant is designed, the size of it is designed to cover essentially the whole macula. In this case, the primary endpoint was safety at 1 year post-implantation, and secondary endpoints were a variety of visual acuity, looking at visual field and retinal function, other exploratory endpoints. The subjects are being followed up, and even for long-term, up to 5 years. And again, there were 2 cohorts of patients, patients who were 2200 visual acuity or worse, so legally blind in the implanted eye, 2200 definition of legally blind or worse. And then a second cohort, patients who had less severe disease, looking at visual acuities of 2080 to better than 2400. Exclusion criteria were essentially no other retinal diseases. So in this particular case, the implant, patients are screened. They're given a low dose of tacrolimus starting 8 days before the implant. The implant takes place on day 0, and they're followed with a variety of different ophthalmologic exams, fundus imaging, a variety of different visual field assessments during the period of time. They're left on tacrolimus for up to 60 days. At day 42, there's a taper. This trial was conducted at 6 different institutions in Southern California, with the principal investigator being Amir Kashani, who did all of the surgeries in this particular clinical trial. The initial results from the first 5 patients in this trial were published in Science Translational Medicine in April of this year. But I'll give you an update on where we are with the trial. We have completed enrollment in this particular trial. We've shown now from the studies that delivery of the implant is feasible and it's safe. We've refined the surgical procedure to bring it down to approximately 2 hours and to work out some of the kinks and procedures that were involved in that insertion process. We've shown that in all cases, the implant was properly placed over the area of GA, and there was a good safety profile with no unanticipated SAEs associated with the implant. One of the things that really excited us is that even in these patients, the first 5 patients that we're reporting on are patients who had 2200 or worse disease. In some cases, it was simply count fingers. But we were able to see reconstruction of some of the layers of the retina after implantation of the membrane. And in fact, looking at the external limiting membranes, again, suggestive of recreating of our retinal architecture, even in these very severely impaired patients. We saw those in terms of, in many cases, people with this visual field are approximately, these visual fields have very poor or no fixation. We've seen the recovery of fixation in a subset of these patients. And then we also see that most of the treated eyes have had either a better impact in terms of visual acuity than the control eye. So we're further following up these patients. Enrollment is complete, and we will be reading out on the whole trial next year. So we are very, very active in preparing for the phase 2 clinical trial right now. So I'll stop there and thank particularly the patients and their caregivers who were terrific during this particular clinical trial, and our various team members, and in particular CIRM, who provided funding for this program, and our partner, Santen Pharmaceuticals. Thank you.